Thanks, David. Yeah, I think that there's a few things that have certainly accelerated the use of cell-free systems in the last few years, and I really think that there's been an absolute explosion of interest. First, as I mentioned, just there's a lot of technological innovations that have led to greater than gram per liter protein yields of model proteins, extended reaction durations, robust and reproducible protocols that other research labs could pick up, essentially democratizing the field and allowing others to enter it. This ability to allow for systems to be freeze-dried, as well as the extension of cell-free systems to genetic networks. So there's been a lot of innovations around using cell-free systems for sensing molecules. Could be sensing nucleic acids, could be sensing small molecule chemicals. And these types of approaches have really been transformative in the sense that it's opened up new application spaces for cell-free systems. And so when I think about why and how and in what ways the field has accelerated, one are certainly just the technical renaissance that has occurred. The second, I think, is the extension and demonstration of genetic circuits being used has been critical. And the third has just been the recognition that time to product, accelerating time to market, perhaps, is really critical. Certainly, I think in the wake of COVID-19 and all that we have faced as a planet over the course of the last 18 months plus, as we look at the worldwide pandemic, it has become more and more evident that having the ability to rapidly iterate through development of protein therapeutics is critical. And I think cell-free systems offer a very powerful approach where you don't have to go through cells, and because of that, the process can be accelerated. And I think that this kind of accelerating time to market, accelerating time to design is something that has really expanded the utilization of the field. Let me give you an example. So just in some recent work of our own, we've been collaborating with David Wiesler, David Baker, both at the University of Washington, as well as Michael Diamond at the University of Washington, St. Louis.
Ah, small world. That was actually our last podcast guest. We had David Baker on last.
Oh, super. Yeah, that is totally small world. And David's been a great collaborator and friend and amazing innovator to work with. And what's been really fun is as we've tried to think about how do you develop therapeutics faster, right? What we found was this interesting area to innovate at the intersection of protein design, cell-free systems, and high throughput experimentation. And so I think, as we're all aware, when you look at antibodies, they have been a very effective therapy in the fight against COVID-19, but are bottlenecked by challenges surrounding kind of the evolution of the spike protein and how variations in spike protein amino acid sequences may change the way in which those antibodies efficiently bind to the virus to prevent interaction with the ACE2 receptor. And so what we were able to work on with David is developing a series of multivalent mini binders, de novo designed proteins that David's group designed, and tune their binding affinities to be quite tight for different escape variants, spike proteins of the virus. And this was really enabled because we could go from rapid DNA synthesis into rapid cell-free expression, into rapid evaluation of binding, and turn that cycle pretty quickly. And to a point where, we can kind of move down into the, days to weeks cycle time now for being able to find very efficient binders to the next potential variants of concern that could emerge. And interesting to note that the current best potential therapeutics that we've identified actually are able to neutralize all of the variants of concern so far that have emerged with respect to SARS-CoV-2 and its variants. So I think from the cell-free perspective, some of the powerful elements, if I just kind of go back, are really this technical renaissance, the kind of ability to make more and more complex proteins, the ability to freeze dry, produce different types of genetic networks, but then in addition, this accelerating time to market has really become appealing for not only academic, but also I think industrial application spaces. Now, David, to your second question, in the context of, what do we still need? What tools are we still currently lacking? I think there are some gaps that still certainly exist in the cell-free space. When we look at glycosylation, for example, of protein-based therapeutics, this is a space that we are continuing to work on in my own research laboratory in very tight collaboration with Matt DeLisa, who's at Cornell University. There are also areas of trying to continue to make these systems robust and work at large scale. While some companies have scaled them, this is certainly not commonplace. It's not common for people to be running 100 milliliter cell-free reactions in their research laboratories, and I think that this would open up some very exciting new opportunities. Last point that I would just raise is continuing to reduce costs. Is there a way to imagine process-based innovations or others that could reduce costs of the system to make them even more accessible? And so those would be perhaps three areas of development that I would be very excited to watch over the next few years as the field continues to rapidly mature. I think the field has emerged already. I think that it is coming out of and into kind of exponential. I'm going to go back a second. I think that the field has not necessarily. I think that the field has emerged already. I think that it is now maturing rapidly into this exponential phase growth, and I'm really excited to see what comes next.